people who knew me and people who knew you were like, you both like science.
It's true. Yeah, fact.
You both like nails and doing your nails. They're like, you two should meet. So hi everyone, welcome back to my channel. You probably all know who I am by now, but you're probably wondering who this lovely lady sat next to me is.
I'm Michaela Banks, and I'm your friend, and hopefully not foe, when it comes to science and nail art. I guess we'll see.
You are my nail art friend.
I am your nail art friend, yes.
Yeah. And you also have a PhD, you're a scientist.
Yes, so my PhD is in molecular biology. So that's really tiny biology. Things are happening inside the cell.
Yeah, so we're kind of the opposite ends of the science spectrum, right? Like you're the incredibly tiny. Yeah. And I am the universe. I am the huge. So like I like how together we kind of cover all the scales of the universe a little bit, right?
Yeah.
So yeah,
or ends of it, yeah.
Yeah. So basically with our love of painting nails and nail art on YouTube, we figured why not film a video where we sit down, paint nails, chat science.
My favourite.
Yeah, obviously.
And nail art.
And then you guys could join in the chat too.
First.
I'm going to paint Michaela's nails first because I think Michaela is a lot better at nail art than I am.
I've been doing it a while for fun.
While I pick nail polishes, and while I decide what colour most resembles molecular biology, why don't you tell the world? Ooh, purple.
Excellent choice.
I carol about you. I'm going for this one. Why don't you sort of tell us like what your PhD was on and what your research is about?
Yes. So some moons ago, I used to investigate cancer testes antigens. Basically, it's a group of proteins that you either only find in the testes or in cancer tissues, kind of anywhere in the body. And that's because the production of sperm has a lot in common with the progression of cancer, how it develops, how it spreads. Like all good researchers, looking at a very tiny part of that and one protein. And I was looking at how this one particular protein was or was not involved in getting messages from the nucleus, where the genes, where your DNA is, out to the rest of the cell, so it could be made into a protein. It's proteins that do the functioning, and DNA is like the template for that protein.
So were you trying to like isolate the proteins that it could be like synthetically made? Was that the idea? Or?
We made it in bacterial cells. We got it out of human tissue culture. Yeah. But basically, we were trying to get that protein and then do certain experiments with only that protein to see what would happen. Right. So we had experiments where, things would go up or down if it was involved in export, basically. Okay. Export of the DNA messages out into the the rest of the cell to be made into a protein. So we referred to these things as licensing factors. You can think of them like a passport. Yeah. So it only allows certain messages to get out, because there's a whole process. Obviously, it's very complicated, but basically there's a whole bunch of proteins involved with carrying these messages through a pore in the nuclear membrane. And it's not passive, so it's not like anything can get through. They have to be allowed through. And so the protein I was looking at, we hypothesized, was one of those kind of passport licensing factors that would let certain things out. Okay,
I'm still confused how this ties in with cancer then.
Cell replication, or cell death, or whether a cell goes, hey, I'm going to try and get out of this tissue and move elsewhere. So metastasis in cancer, so when it spreads. All of these things are basically controlled by whether or not certain genes are on or off. And so if a gene message can't get out of the nucleus, it's off. Right. Whereas if it can, it's on. So the idea is that this passport was allowing messages for essentially bad genes to be switched on, and so it could be saying, hey, you should now replicate crazily and cause a tumor. Right. Basically.
Okay. But particularly you were focused on testicular cancer.
No. So
it was testicular cancer.
No. So it's normally only found in the testes. So under normal conditions you would only find it in the testes. Okay. if you found it in any other tissue, it could be breast, colon, skin, whatever, then it was a cancerous tissue.
Right, yeah.
So the only time we found it not in the testes was in a cancerous tissue.
Right,
but it was found in, I think it was something like 15 to 20% of all different types of cancer. But the idea was that if it did something like that, I've just done a smudge.
No!
Sorry.
Smudge.
Sorry, it's still looking good though. Look, it's just here.
Oh, okay.
But the hope is if it was sort of involved in that sort of process, then if you tried to target that protein with a drug, say, to just turn it off or get rid of it, then it would stop that cancerous protein.
Right.
So, and the thing is, for women that's fine but we didn't know what it was normally involved in when it wasn't in cancer.
Right. We should do top coat while you're talking to me. Which one's top coat?
That one. Yeah, so you'd be able to sequence their genome, maybe looking for particular markers, and you'd be able to say, is this a LUSP4 positive?
Oh,
so LUSP4 was the protein I worked on. Leucine zipper protein 4 is a very exciting name. But yeah, you'd be able to say, is this a LUSP4 positive cancer, and therefore should we target that, or is it a something else positive?
Should we, I don't know how long this top coat takes to dry, but I'm excited to see what your like nail stamping things are, because I've never used these things before, so I'm really excited.
Oh, I can talk you through this.
Oh my gosh, look, they've got like DNA and like elements from the periodic table. They've got all like the flammable symbols and like the radioactive symbols. Also, there's also like some pills. You're taking you can have I want to keep calling it Les Paul. I don't know why. What is it called?
LUZ, LUSP4.
LUSP4. It does help knowing how to spell it. So I think I'm going to go for some pills and the cute little DNA molecule. Why? What did I do?
Because that DNA isn't strictly accurate.
What?
No.
Why not?
Well, so we all know DNA is like a twisted ladder, right? But like
the double helix.
I'll have to draw it. Can we see
we can
here?
see it here. This is like double use
exciting,
paper, right?
Yeah.
It can be used as drawing and for nail polish.
That's one strand of DNA. So what I mean is it looks like that. And what they've done is I can't even...
Come down, yeah.
I'll come down. So what they've done, I can't even do it because it's like, so they've
done it really broad.
Well, and it crosses equally. So you see here, that is a minor groove.
Right.
And that is a major groove. So that is a small bit and that is a large bit.
No, I don't get it.
So here, can you see that both of these are the same size?
As in,
do I need to do it even bigger?
Indents,
the indents.
So the indents are the same size, right?
On that one.
Oh, I see. So this indent's bigger, and this indent is not as big,
one.
right? I just thought it was the way you'd drawn it, like by accident.
No. That's what it looks like. And the other thing is, it has to twist the right way because there's this thing called chirality, which is the handedness of molecules.
I remember learning about that like. Caraway seeds and mint are like the same molecule, but they're like reflected, so they have completely different flavors.
Yeah. So the right way is clockwise, so it kind of turns like that.
So like the whole helix molecule winds clockwise, right? Okay. And
exactly.
then on the plate,
it has no handedness really.
It doesn't, does it? I'll tell you what. I'll do one with a stamping plate.
Yeah.
And I'll do one freehand.
Okay.
Scrape, scrape, scrape.
Yeah. Just one good time will do it. Yeah.
That should do it.
That should do it. And then
really? Because I'm going to pick all those things.
Roll it on.
Got to see my first ever stamp.
Da, da, da.
Do I roll it onto the nail too?
It's quite soft, so you can push it.
Okay.
So
just so exciting.
I'm excited. Moving vicariously through.
I don't know how hard to push.
That's it. That's fine.
Look at it!
Da.
DNA on the nail, DNA on the nail. What were we talking about? Oh yeah, the chirality of DNA. Let's talk more about that, because as fun as people think nail polish is, I'm sure more people want to hear about chirality of DNA and how cool it is. So if it's like, if DNA is the left-handed molecule, in the chiralityness, like what is the right-handed molecule that looks like DNA but it's the mirror image?
It's still DNA. I think they refer to them as like DNA A and DNA B, I think is. But I think one is biologically relevant because it's, the version we actually get.
Yeah.
And the other is not. Oh, it smells like esters. Thinking of chemistry.
Should I sniff that?
I don't think it'll kill you. I'd hope they wouldn't sell it. But, there could be like a mirror image Earth or universe out there somewhere, perhaps, where just because on our planet everything appears to mostly be the kind of left-handed version.
Yeah. Kind of like we're carbon-based, but they're silicon-based, and maybe their DNA is the right-handed, DNA strand.
Yeah. Yeah.
Oh, yeah, have a look at my pill that I drew.
It's good. Look at that.
See?
Isn't that beautiful? It's not looking anything like a Pokeball.
I might make it into a Pokeball, though.
Yeah, I'd like, you know how sometimes people do these projects where it's like the genetics of superheroes or something? If someone done the, like, because I've seen people do, like, evolution and Pokemon.
Right.
But I wonder if someone specifically, like, looked into the genetics of Pokemon.
Oh, maybe. I would watch that if they had. Please tell us in the comments if someone has done the genetics of Pokemon.
Maybe you've done the genetics of Pokemon.
Or, like, the evolutionary biology of Pokemon. Whether we could go from current species on planet Earth and somehow get to an electric mouse. I'm now going to attempt to freehand a DNA molecule. So, I want to make sure that I get these, like, little, like, big major troughs and minor troughs. That's what I'm calling them, troughs. That's not what they're called, though, right? Dips?
Let's go with that.
Troughs? What are they actually called? So I need to basically draw, in my head I'm drawing a sine curve and then a slightly out of phase sine curve.
Slightly, yes. But not completely.
I'm shaking. Okay, do you want to tell me something cool about DNA while I draw it?
It's super small, unlike this.
Shh, I'll make it work. If not, you'll just have a really cool, wiggly nail.
I mean, that is nail art right there. Yeah. Look at that. There you go.
Right, now I have to...
We've got one strand.
You're telling me I have to now do an out-of-phase sine curve on top of that? Do I have to then join them up with the...
I mean, you could do the little rungs of the ladder thing if you want.
They have letters, right? Like, I've always wondered why those letters were picked. Like, is it
It's the name of the chemical. So A is adenosine, thymine, guanosine, cytosine. Mm-hmm. Yeah, A, T, G, and C.
A, T, G, C.
And A always pairs with T, and G always pairs with C.
That looks like it's got major and minor.
I think this bit up here is looking pretty good. Yeah,
there's that one there.
And this one is great, and then it goes a little bit like meh.
Well, that one at the end is okay, but then that one in the middle is not great, I don't think.
No, but it's pretty good.
I think I did better than the nail stamping plate. I mean, not to toot my own horn.
You did 50% better than...
50%. I like that it's... I gave it a number. I am 50% better than a professionally printed nail.
Yeah.
Should I try and join them with
go for it.
Now? But how many do I put per sine curve crossover?
I think there's... Oh, God, no, there is an actual number. You should have told me I needed to bone up on that.
I'm just so interested in
like
DNA.
10 years ago. I think there's like 2 point something base pairs per turn, maybe, something like that.
So yeah, so this was so long ago for you because you don't do this anymore, right? No. You don't do research. So would you like to tell the people what you do?
I help researchers engage with the public. And I do that across what's called the Maths, Physical and Life Sciences division at the University of Oxford. So I actually get to hear about things in physics, engineering, computer science, zoology, plant sciences, stats, like so much stuff. Sorry if I missed you out. Chemistry.
Yeah. Oh, that's a freehand. I've never done any freehand nail art.
There you go.
I think that's pretty cool. Hollow top coat or top coat?
Oh, if you get a hollow top coat, I want a hollow top coat.
Well, everybody loves sparkly, hollow DNA strands, right?
Exactly. If you're like glamorous DNA.
Yeah. Can you promise me something? If you ever like get back into research and you start designing the form in which people would take your Les Paul drawing. Yeah, Les P4, was that what it was? Then can you design it in a strange Pokeball pill form?
Yes.
Just like this on your nail with extra like edible glitter.
I'll do my best.
Is anybody watching who works in the treatment of cancer via oral medication? Please can you design some pills that are Pokeball shaped for our joy.
Our joy indeed. And the joy of patients.
I'd never throw them out. Maria Kondo would be like, does that bring you joy? And I'd be like, yes, it does. Yeah. We should paint my turn.
Let's paint your nails. Okay, well. So, galaxy, so obviously space, right? Mm-hmm. So it's going to have a black background.
Yeah.
But what would make galaxies kind of identifiable? What different shapes do they come in and not come in? And like, how can I best do this for you?
Okay. Well, it's funny because there's not actually like an agreed on definition in the field of what a galaxy is.
Really?
Yeah, it's weird.
So not just like a massive collection of stars.
That's basically the closest we've got.
Right.
Because it's very difficult to define because there's like stars, there's gas, dust, dark matter, there's a black hole in the middle.
Right.
So I was thinking when you picked black, right? Like, and you were like, oh, space is black. Right?
Isn't it?
Well, yeah, well, that's the thing. But the like, someone once asked, and it was like called a paradox for a while, but it was one of those paradoxes, it was just because we didn't have the knowledge to understand why it was a paradox. It's called Olber's paradox, right? And it was like, he asked, why is space dark?
Yeah.
Right? Because if space is infinite, then every line of sight, like every single point that you look at in the sky, eventually you should like hit a star.
Yeah.
Eventually you'll happen on a galaxy or a star or something that has light. And it's weird, therefore, that the sky is dark. Because if there's stars everywhere, because the universe is infinite, then it should be light. But then obviously we learned that the universe isn't infinite. So space is black because the universe is finite. Messier 87 is the biggest galaxy. One of the biggest galaxies. Yeah. And it's got this massive big jet of emission coming off from the black hole. So it's like a big yellow blob and then this big like bluey purple like jet that's like bigger than the galaxy itself that's going from the black hole.
Is it globular?
It's globular.
It is globular.
Yeah, do you want me to draw it?
Yeah, draw it for me.
So in a lot of images it's just like blob of a galaxy, right? And then it's just like jet.
Just one jet?
Yeah, and
like
the
a
jet's
tadpole.
all like...
Okay, and then this bit you said was like yellow...
Yellowy, yeah, like yellowy and just glowy.
Uh-huh, and
rather
then this
than,
bit is bluey...
Just bluey purple.
Bluey purple, right.
Rather than like, you know, like a spiral galaxy would be like this. You know, like barred spiral like that, and then the whirlpool is like proper, like, you know, full on like extra arms coming out. And,
wow.
So.
Oh, okay, well I'll start with this one.
M87 is really, really old, and it's sort of like not really forming any stars anymore. And
right.
So it's full of a lot of older, smaller stars, which are sort of at the end of their lives, but will just sort of continue burning because basically the bigger you are as a star, the quicker you're going to use all your hydrogen fuel. Right. It's like live fast, die young kind of thing for stars, like
Kurt
the rock
Cobain's
stars, right?
of
yeah, exactly. Also, it's like the center of like the local group of galaxies that we live in. So there's like the Milky Way, right? Is like our galaxy, big spiral galaxy, and then there's Andromeda, which is like nearby us, and for folks in the southern hemisphere as well. Maybe we have Australians watching, New Zealand, I don't know.
Hey, howdy. No, good day. Sorry,
G'day.
one.
In the southern hemisphere, you'll see like there's the small Magellanic Cloud and like the large Magellanic Cloud as well.
Yep.
Which are like little dwarf galaxies in the Milky Way. And then that's like one group of galaxies, which is like the local group. But then that group of galaxies is in like a cluster of groups. And the very center of all of those things, in like the gravitational driving seat of the center of all the galaxies, is M87. Because it's like one of the biggest.
So yellowy orange, you say, for the bulgy bit.
Mm-hmm. And then like bluey purple bit.
Okay. There we go.
Yeah, that looks about right. Yeah, that looks great.
Is that alright?
Looks brilliant. I got a t-shirt from M&S once that had like actual space images on it, like it wasn't fake data, and I was like so happy. And I tried to put it through this software that astronomers use that can tell you where in the night sky you are based on like all the stars.
That's cool.
Yeah, because like basically what it does is it draws triangles between all the stars and then tries to work out like which triangles then that matches up with.
Yeah.
And yeah, like I was like, it's so going to be able to do it, but I think it was like distorted on the t-shirt. Like it had been a real data, but they'd like stretched it a bit or something, and so to like fit the dimensions of the t-shirt, and it didn't work. And I was really sad.
Bummer. But at least they like used the real thing in the first place. Yeah, that's pretty awesome.
But you know that software I was talking about? It's called Astrometric Net. If anyone has like images of the sky they want to play around with, but that same software, like that algorithm, is now used by marine biologists to identify whale sharks.
Oh,
yeah,
that's cool.
It is cool, isn't
it?
It's
like in the water.
Yeah, like in the water when they see them on the videos. Because apparently whale sharks. I mean, I'm not an expert, but apparently whale sharks, like they have those dots on them, right? Like they're like big blue things.
Right,
they have the white.
Identify the individuals.
Yeah, exactly. Because they have the white dots on them, and apparently the white dot pattern is like individual to like each whale shark.
That's awesome.
And so like you can use that to identify and like track them without putting any like tracking things on them.
That's pretty awesome. Shall I give an attempt at the Whirlpool? Sure. So can I draw it like that?
Yeah, draw it like that.
Okay.
Right. It also technically has a little companion as well that's like a blob over here. But we can skip that. You could be like, it's out of frame because my nails are so small.
We'll see where we get to. This is why I have like mahoosive claws, just because then
so
there's more
space.
What
if it's like a deer?
It's like a deer.
A bit of deer on it.
I think the main thing when you're doing this is knowing when to stop.
Yeah, you've added enough.
Yeah. It's like, I could just make it just a little bit.
That's good though. I like how the arm stops on the edge of the nail.
Does this thing need to be a colour? Is it blue? Because like Whirlpool.
It's bluey, yeah. It's bluey, pinky.
So it's relatively young?
Yeah. Young.
Yay, I'm learning things!
So like the pinky areas are hydrogen and then dust in the galaxy that sort of like make dust lanes on the galaxy as well. But there's also lots of star clusters in the Whirlpool Galaxy as well. So like big sort of star-bursting regions where there's like lots of stars forming in a very concentrated area. And like when you see those, you can almost see the individual like massive stars that have been formed. And obviously like reason it's pink is because hydrogen sort of glows in pink, and hydrogen's great because hydrogen is like what you need to make new stars. It's like star fuel. Can I have a stampy plate maybe?
Oh yeah. Oh, what one do you want? Oh, of course.
Because it comes to this one in the corner. Look, this one here.
What's 3? Like lithium?
Lithium, yeah.
Is it a lithium atom?
Well, there's 7. So I'm assuming there's something hidden behind that I can't see on the other side. So let's say it's beryllium.
Okay, beryllium.
But it's got 3 electrons. So it's like a beryllium ion.
Yeah. Do you want pink?
Yeah, pink sounds good. Stampy.
There you go.
Oh, yeah. Look at that. I have an atom on my nail. I'm imagining this is like the early Big Bang, and all that there is in the universe is lots of hydrogen, lots of helium, a tiny trace amount of lithium and beryllium. Here is the trace amount of beryllium. Like most of the stars are in the spiral arms, right?
Yeah. So that's where the most intense. But it's not
like there's no stars in between spiral arms, because like we don't actually know, we don't have an accepted theory yet for like how spiral arms form and like what they actually are. Yeah, so there's a couple of different like ones where it could be. So one of the most popular is like it's like a density wave.
Right.
So you can imagine like a flat disk of stars, and there's just sort of like a wave of density like moving through the disk that like pushes all the stars together to make it look like there's a concentration there. You know, like a Mexican wave?
Yeah.
Right, is like not an actual...
No, oh right, okay, yeah, because it's people moving in sequence.
Moving up and down, but you see it as something moving around the stadium, right?
Yeah.
But there's nothing actually physically moving around the stadium, because all people are doing is standing up, like moving up and down in sequence. Like you can imagine it like that, the spiral arms are just the points where people are stood up. I feel like this looks like some sort of superhero logo rather than a galaxy, but I can't decide which one it is. It reminds me of something. If you can think what that superhero logo is down in the comments, please let me know.
Right, let's get you some stars. I'm just putting them on randomly.
I like the sound effects of like the creation of a star.
And look, I've managed to like paint my hair as well.
So when you think about a star, and people see lots of images of stars, they're obviously not round, and they're not like, you know, a 5-pointed star shape either. They have like these, what we call a diffraction spike, right?
Yeah.
So there's like a line like going in a cross basically through the center of the star, and that's basically when the star is so bright that there's so much light coming from that star that it's hit the detector, and it's hit like a couple of pixels on the detector, and that pixel is like completely full with light very quickly, and they sort of like bleed over into neighboring pixels. And so, despite the fact that it looks incredibly pretty when you have these like huge,
that doesn't look very pretty.
It does look pretty, like diffraction spikes on the camera. Like that's what it is. Basically, it's an overexposure, and people tend to try and avoid that as much as they can. Like when you're using a telescope, you try and pick the exposure time to be appropriate for what you're viewing.
It wouldn't be galaxy nails without it, though.
That's very true, because when you observe galaxies, that looks awesome, actually. Look at the diffraction spike on the star. Like, when you observe a galaxy, though, say I'm observing M87, but there's a star in the foreground, like, that star will be much brighter than the background galaxy, which is much fainter. So even if you picture exposure so that you get enough light to be able to see the galaxy, you're always going to overexpose the star in the foreground, and so you can end up with big diffraction spikes.
Oh, look, here you go. This can be a star in the foreground.
Yeah, do it. I love how long we've been sat here that it's gone dark.
Has it? What time did we actually start? How long have we been talking for?
I think we started, well, this can tell us, we've been talking for an hour and a half.
Whoa.
We should talk about the science of hollow next. I mean, it's not that difficult, but still, we should talk about it next time.
Or what about the people in physics? What do the quantum rainbows?
Oh, yeah, we should get
them bob, because it's not strictly hollow, but...
Hollow talk.
And in that light as well, it looks amazing. There we go. So is that a pleasingly, vaguely accurate galaxy now?
It is. Yeah, we've got M87 with its jet over here, which is beautiful. We have the Whirlpool Galaxy complete with companion. We have Random Barred Galaxy that also kind of looks like a superhero logo.
It does,
but we can't remember what. And then we have a little bit of beryllium on this thing, which is beautiful.
Why not?
So we have DNA, a Les Paul pill, my hand-drawn DNA.
Yeah, which is amazing.
But yeah, like, if we get guests on, we're going to do a guest's hand each, aren't we? Like, if we get scientists. And then we'll get them to pick which one their favourite is. Like, you'll do one hand, I'll do another hand.
Competition.
But this time there's no guest. So I think that we should ask people to vote, which is your favourite? Is it the DNA and pill nails that I did? Or is it the galaxy nails that Michaela did? This was the very first Nailing Science. We hope you enjoyed it, and we'll be back in a couple of weeks, hopefully interviewing another scientist while we paint their nails unsuspectingly.
Bye.
Bye from us, and bye from our nails. Bye.
I'm gonna get this wrong, aren't I? DNA should
We'll record both, and I'll edit it later. We'll just pick one. How did the YouTuber beauty gurus do it?
They like do it from above or something. So that could be a citizen science project, like Galaxies. Galaxy. What did I say?
Did I say
Galaxy?
Galaxy Zoo in the universe, but I like it. I'm going to start calling it that now. Welcome to the Galaxyverse. Scientists are obsessed with archiving, right, and making sure that no data is lost, no writing is lost, or anything like that. You know, we should start the Scientists for Archiving Nail Art. Okay, well, don't sound thrilled. You know what I always wonder is, what other knowledge could I hold in my head if it wasn't for all the song lyrics I know?
Yeah, so if you got rid of, Backstreet's Back, you could
I'd never get rid of Backstreet's Back.
Well, no, that's important, very important.